Welcome to this channel for biology. Today we are going to deliver a brief lecture on aneuploidy and related genetic disorders. What are actually the aneuploidy and why the aneuploidy cause the genetic disorders, dangerous genetic disorders? What are the different genetic disorders developed as a result of aneuploidy? So today we shall discuss about aneuploidy and related genetic disorders. Aneuploidy is a phenomenon where the chromosome numbers in a gamete is reduced or increased than the normal haploid number of chromosomes, so that they can either develop n+1 or n-1 gametes. As a result of this, after the fusion, the new individuals develop with one or more chromosome numbers, increased chromosome with one or more increased chromosome number or one or more less chromosome number. Sometimes the increased chromosome number may be the sex chromosomes or sometimes the autosomes. So as a result, the abnormalities developed. In this case, we are first discussing about aneuploidy. What is actually happening here? Aneuploidy. Here it is looking here the human cell where 46 number of chromosome is there. I have not here the showing 46 number of chromosome. Here only 1, 2, 3. 3 pairs of autosome I am showing and one pair of sex chromosome is showing is the female cell. So as you know that the female egg is a main arrested up to the diploidine stage of prophase 1, and then after the entry of the sperm into the primary oocyte, the primary oocyte will divide and secondary oocytes and final egg will be developed. So in this case, what happened with the aneuploidy? In a female cell, 46 number of chromosome is there and each chromosome is already divided longitudinally to form a 2 chromatids. In an anaphase 1 of cell cycle, what happened during the meiosis? Each chromosome remains divided. Each homologous pair, this is a homologous pair, these are homologous pair, these are homologous pair, and each chromosome is divided longitudinally. So it's no problem. In anaphase 1, we know that from the center, it is attached from the centrosome. The spindle fiber attach with the centromere region, so that proper chromosome segregation takes place. If the centromeric regions are not properly attached with the spindle fiber, so that chromosomes will not equally divide into 2 cells. Here the single cell and chromosomes are attached with the spindle fiber to the centromeric region, each chromosome, except this one. As a result, the chromosomes are not equally divided into 2 equal halves. Here the 1, 2, 3, 4 pairs, so that after a cell division, each 2 cell from this cell, 2 cell developed after anaphase 1, 2 cell is developed and 2 cell half number of chromosome is developed. Here the 8 chromosome is there, 4 pair. So after first round of meiotic 1, this is a meiosis 1 division. First round of meiosis 1, each cell will contain, it must contain 4 number of chromosomes and 4 number of chromosomes. But here you carefully notice that this chromosome is not attached to a spindle fiber. This is the main cause of chromosomal non-disjunction. This is the main cause, that is the spindle fiber is not properly attached with the centromeric region of a chromosome. As a result, these chromosomes lag behind and not going to this pole, As a result, what happened here? From the single cell, two cells is developed with unequal chromosome numbers. This is a 1, 2, 3, 1X chromosome, 1, 2, 3 and 1X chromosome. And as this chromosome is not attached with any spindle fiber, this chromosome lag behind and remain in this side. As a result, this cell contains one extra copy of chromosome with two chromatids. In this case, this cell should have 4 chromosomes, 1, 2, 3 and 1X chromosome, but here one chromosome is absent. One chromosome is absent. Here one chromosome is extra, here one chromosome is absent. So as a result of first meiotic division, two cells is developed, one with n plus n plus 1 and another with n minus 1. One with n plus 1, another with n minus 1. One with n plus 1 and another with n minus 1. So in this case, it is clear that after the first round of division, the chromosome numbers is reduced to half and unequally reduced. Due to what? Due to this chromosomal non-disjunction, that is, a spindle fiber is not properly attached here. As a result, this chromosome lag behind and the two unequal cell is developed with unequal chromosome numbers. And after second round of replication, we know that the second division of meiosis, NFS2, second meiotic division is like a mitosis division. As a result of this, what happened here? If it is normal, then it is normal division occurred. Then what happened here? 23 plus x. This cell will develop two cells, 1, 2. So in this case, here 46 chromosome, half it is reduced to 23. So here the 23 plus x. So what happened here? We know that the 46 number of chromosome, out of the 46 number of chromosome, 44 are autosome and 2 are X chromosome. So as a normal rule of meiosis, what will happen? 22 will be there and 22 will be there with 1 X chromosome. But in this case, what happened? 23 is here, one extra, n plus 1, 22 not, 23 is there with one extra X chromosome, and in this case 21 is there, as one chromosome is not going here, it's going to here. So by instead of 22, it is represent 23 chromosomes, and here 22 minus 1, that is one chromosome is going here, so 21 number of chromosome autosome is there and one X chromosome is there. So after the second round of meiosis, what will happen? It will develop equal number of chromosomes. It is a mitotic-like structure, mitotic-like cell division. We know that meiosis 2 is a mitosis-like structure. So why the gametes produced from this cell is, after the meiosis 2, is 21 plus X and 21 plus X. These are the reduced, that is n minus 1 gamete, n minus 1 chromosome. This gamete contain n minus 1 and this gamete contain n plus 1 chromosome. So these are the problem with the chromosomal non-disjunction. This is a common example of aneuploidy. Another important thing is that in this case, chromosome number is reduced but not becomes half, as due to chromosomal non-disjunction. So this is the chromosomal non-disjunction. Now we shall see that what problem we will face due to this chromosomal non-disjunction. In the next page we are going. So what happened? Another slide is there. Chromosomal non-disjunction or chromosomal non-alignment in this case occur at the anaphase 1 stage. So why? All 4 chromatids, all 4 gametes are affected. But if chromosomal non-disjunction will occur at the anaphase 2 stage, here the anaphase 2 stage, and the anaphase 2 stage, in this stage is a normal, that is 46 chromosome equally divided into 23. But after this, at the anaphase 2 stage, during the separation of chromatids, here the chromosomal non-disjunction occurred. That is, the spindle fiber not attached with the properly attached with the centromeric region. As a result, what happened? This chromosome pair will go to this site. As a result, unequal number of chromosomes, two cells will develop with unequal number of chromosomes. In this case, here the 23 plus X and 21 plus X. Why? Normally, what will happen? Normally, it is normally if proper alignment of the chromosome or proper cell division will occur, then what will happen? 22 plus X and 22 plus X. This type of gametes will be produced. But in this case, what happened? In this case, due to the unequal division of these two chromatids, these two chromatids are not going properly in those both poles. This totally going to this pole. As a result, 22 instead of 22, 23 plus X. This X, 23 plus X and 21 plus X number of chromosomes. As a result, 22 here number 1, n minus 1, and in this case n plus 1 gamete is produced. As a result of this, only half of the gametes are defective, but in this case it is normal. So when when chromosomal non-disjunction occur at the anaphase 1, then 100% gametes or all 4 gametes will be defective, and when chromosomal non-disjunction will occur at the anaphase 2, then only 50% gametes will be affected or will be of different type, or they will be of abnormal type. So in this case, here the example here. Now what will happen? Here the sperm, here is the egg. This sperm contains n plus 1 gamete, and this is a normal egg. If this sperm contains 1 plus n plus 1 gamete, that is, it contains 20 instead of 22 plus X, it contains 23 plus X. This extra chromosome is due to chromosomal non-disjunction. And here look at the first slide. Here, suppose here the 23 plus X. This 23 plus X, suppose it is a male cell, and 23. This is a 21st number of chromosome. As we know, this is in Down syndrome. In Down syndrome, 21, that is the 21st pair of chromosome in human being contain trisomy, that is 3 copy. 21st number of chromosome, homologous pair contain 3 chromosomes. So why? This is a trisomy. So in that, as a result, the sperm, suppose it will produce n plus 1 gamete and egg is normal. So what will happen? Then 22, 23, 20, 45 plus double X. That's a, which is called Down syndrome. That is, where is present extra chromosome? Extra chromosome is present here, the 45. Where? Now 21st number of chromosome, the 21st number of chromosome is trisomy. Instead of presence of 221 number of chromosome, in this case 23 number, that is, the sperm contain 221 number of chromosome. As a result of this, the trisomy occurred, and as a result of this, the problem arises. That is Down syndrome. In case of Down syndrome, chromosomal trisomy, that is, sperms contain n plus 1 and egg contain n number of gametes. Why n plus 1? That is, a plus 1 is due to the 21 number of chromosome. That is 21 pair, that is 21 first pair of homologous pair contain extra chromosome. That extra chromosome, due to the presence of 2 copy of 21 number of chromosome, 21 pair, 21st pair, and in this case is a normal 1 pair, 21st chromosome, as this gamete is normal. So why in this case 3 copy of 21st chromosome is there instead of 2. So why it is called Down syndrome? We know that Down syndrome contains so many abnormalities in individuals, like retarded brains, height short. These are Down syndrome the example, and abnormal mental situation, improper gonadal development. So all are Down syndrome examples. Now the aneuploidy, another example of aneuploidy here, where if aneuploidy occurs in such a way that X chromosomes are not properly aligned with and non-disjunction occur with the X chromosome. That is, during the anaphase 1, out of the two X chromosome, if only one X chromosome is attached with the spindle fiber and one chromosome remain left, so why this extra X chromosome will go in that direction and not equal division of the X chromosome will occur. So as a result, this cell will divide unequally and produce, what will produce? The two cell, they will produce two cell, one with both the X chromosome, another with no X chromosome. So after the anaphase 2, what will happen? They will develop 22 of autosome plus 2 extra X chromosome and 2 extra X chromosome, and in this case 22 number of autosome will be okay, then no X chromosome will be there. So all 4 gametes will be defective and one gamete will be of 2 gamete will be of n+1 and 2 gamete will be n-1 type. So as a result, what will happen, what abnormalities will develop in this case? Here the sperm is okay, suppose n, sorry, in this case double n is present, that is the n, 22 plus y is the sperm chromosome number is the normal sperm, and eggs contain abnormal egg, that is 2 X chromosome is present in the egg cell n-1. As a result of fusion of these, 44 plus double X plus y, that is 47 number chromosome. This abnormality is called Klinefelter syndrome. This Klinefelter syndrome is due to aneuploidy occur during the segregation of chromosomes during the meiosis, and that occurred in the anaphase 1. And if X chromosomes are not properly attached to the spindle fiber and not equally divided into two opposite poles and into the two different cells after the first meiotic division or during the second meiotic division, maybe as a result unequal number of X chromosome will go to different gametes. As a result, abnormality is developed. So as a result, in this case, what happened? 47 number of chromosomes. This is called Klinefelter syndrome. That is, the female will develop with some abnormal male characters. Female will develop baby with female and will develop some abnormal male behaviors, male characters with secondary gonadal structures, secondary sex organs. These are Klinefelter syndrome, and this syndrome will develop due to this gamete, n+1 gamete. That is, when the gamete, the egg, where both the X chromosome is present, but the gamete is also developed here. Look here, this gamete with no X chromosome. What will happen with this? If these gametes will fuse with a normal sperm cell, then what will happen? Then 44 plus 22 plus 22, 44 plus X plus 0, that is 45 number of chromosomes will be developed. As a result of 45 number of chromosome, when the 45 number of chromosome is developed in this case, what will happen? This is called Turnout syndrome. In case of Turnout syndrome, chromosome number is reduced to 45 instead of 46 due to the chromosomal non-disjunction during the meiosis, and one gamete, one female gamete especially, or male gamete may also be possible, whatever may be that is unequal distribution of these sex chromosomes. So why the Turner syndrome developed? So as a result of unequal distribution of chromosome, unequal segregation of the chromosome during meiosis due to the chromosomal non-disjunction. Chromosomal disjunction is the term that is used when proper chromosomal segregation occur, and chromosomal non-disjunction term is used when chromosomes are not properly segregated into opposite poles. So why it is called chromosomal non-disjunction. So this is the overall phenomenon that we have discussed here, that is what is Down syndrome, why Down syndrome developed, why chromosomal abnormalities cause Down syndrome, and why chromosomal abnormality is called Klinefelter and Turner syndromes. So this is a overall phenomenon. Thank you very much.